Hello, everyone, and welcome to today's live broadcast, Comprehensive Variation at Clinical Population Scales. Will Salerno from the Human Genome Sequencing Center at Baylor College of Medicine will be the speaker for this event. We are excited to bring you this educational webinar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars for advancing scientific collaboration and learning. This webinar is sponsored by DNA Nexus, the leader in cloud-based biomedical informatics and data management. DNA Nexus enables the advancement of biomedical discovery through the sharing of tools and data sets in a secure, compliant, and collaborative ecosystem. Our work accelerates scientific insights in the healthcare and research space to solve the largest challenges in the market today. I am John Didion, a principal scientist at DNA Nexus, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Q&A button located in the bottom of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. And now I'd like to introduce today's speaker, Will Salerno, Director of Genome Informatics at the Human Genome Sequencing Center, who will share his experience and insight developing a scalable informatics model for at-scale WGS. Please join me in welcoming Will Salerno. I will now turn the presentation over to him.
Great. Thanks, John, and thanks everyone for joining today. Really glad to have the opportunity to speak about comprehensive variation at clinical population scales. This is work that we've been doing here in Houston at the HGSC and with our partners all over the world, but especially at the UT School of Public Health. And for those who might not be familiar with the Human Genome Sequencing Center here in Houston, we just celebrated our 20-year anniversary. We have 180 staff, a huge sequencing fleet of all kinds of different platforms. It's a long history. I've been at the Sequencing Center for about 6 years. But one of the big advances we've made in the last couple years is the launching of the HGSC+, our clinical laboratory. And everyone should feel rest assured that it was safe during Harvey last year. I rode my bike into the medical center, and this is the picture I took before I rode my bike through that, and that was a mistake. But we have survived, and we continue to push here in Houston with all of these partners that we have here. Our vision for bringing genomics into medicine and really driving, this idea of personalized medicine. So, I run the Next-Gen Sequencing Informatics group. We do all the data processing and the informatics R&D for both the research and the clinical sides. So when I think about how we drive research and clinical research into human disease with genomics, I sort of broke it down into this chart, which is by no means complete, but it does sort of lay out the different types of steps that you can have when you're doing genomics experiments that's relevant to human health and disease. And just to go through sort of briefly, you can see on the left that if you start with DNA sequencing, you do whole exomes in a trio, you call your small variants and coding regions, and you compare that to traits of interest. That's what, quote unquote, I'm calling sort of the Mendelian model, or maybe those traits are rare diseases. As you start to expand out into cohorts, which are the green lines, you're still using DNA sequencing. Maybe it's exome, maybe it's genome, maybe it's chip, but you're using multiple samples. A lot of times you still bring that back to small variants in the coding regions. You compare with phenotypes and traits, and that's sort of the GWAS model. Now, obviously, none of these descriptions are complete, but the idea is that we're progressing increasingly right and filling out this graph. Really, I think where we are at right now at the Human Genome Sequencing Center is a lot of data being driven by our whole genome sequencing program and large cohorts. But not limiting that analysis to small variants in coding regions, but also looking at complex structural variation looking at non-coding and regulatory regions, and then not just thinking about, endpoint traits, but also end of phenotypes, integrating longitudinal data, and doing a really robust genotype-phenotype analysis. Some of our forward-looking work is really moving that top row across from DNA into this multi-omic space, with the idea that we really have many ways to navigate the space of human genomic data, with the point being, how do we drive these different combinations of data and experimental designs really into clinical interpretation? How do we make personalized medicine relevant to every individual? This N of one idea. So that's sort of the model of how we navigate this space. The road to this type of clinical population, transitioning from these research to this clinical population, a population of N of one, the road here has gone through a bunch of acronyms, all of these great programs that we work with through the various NIH programs, NHLBI, NHGRI, the NIA. Some of you may be familiar. I'm not going to go through all of these in detail, but I do want to give an overview of the type of data we're dealing with. The TopMed program, is currently about 130,000 whole genomes and a lot of cardiovascular related cohorts with a eye toward generating multi-omics data. That's really interesting. The CCDG program, the Centers for Common Disease Genomics. This is currently about 55 to 60,000 whole genomes that we're working on right now. Multiple cohorts like TopMed, multiple sequencing centers, with the idea that we want to marry the sequencing generation with the specific disease research. Working with the CHARGE and ADSP, the Alzheimer's Disease Consortium, slightly different types of data profiles at different scales, but the idea is that we're working on specific diseases and really going after a well-curated collection of phenotypes. One of the big components in all of these programs is going after multiple ethnicities. Again, this idea that we want to make personalized medicine relevant to every individual. And that means, any ethnic background, we're creating appropriate models for specific subgroups. That's all large research work. The clinical work, a lot of that has been driven, especially by our involvement in the eMERGE program, which is a custom panel of clinically actionable genes. We've sequenced 15,000, the whole set's about 25,000. And this is work done by our clinical lab, the HGSC+UT, where we're doing clinical sign out and really focusing on this idea of how do you automate at a large scale the return of results. So we had these large scale research programs. We had this, very successful eMERGE program where we're doing our sign out. And where this culminated was, over in the last several months, we were awarded one of the Genome Center grants for the All of Us program. Which is, go and sign up online. The goal is to sequence 1 million samples over 5 years. We're doing this with our partners at CIDR at Johns Hopkins. And this is whole genome in a clinical setting with then, validation of actionable variants and return of results. So that's really, how all of these programs that we've worked with has taken us to the point. And it's not just us. We work with so many other sequencing centers and collaborators that this is the road the NIH has sort of been on to get to clinical populations, and we're very happy to be a part of that. A bit more specific to our Houston history, at the HGSC and the UT School of Public Health, we're driving this through comprehensive variation. The idea that, again, when we talk about making personalized medicine relevant to every individual, it's not just about your age or where you live or your ethnicity. It's also about what kind of variants do you have. We want to make sure personalized medicine isn't just relevant for people who have small variants inside coding regions. If you have something complex or large in a regulatory region or outside what an exome or a panel might pick up, personalized medicine needs to be relevant to those individuals as well. So as you can see, I'm not going to go through all of these small pictures, but a lot of this work, especially on the informatics and analysis side, was done in collaboration with our partners at DNA Nexus. And it started with a personal genome, doing a high quality personal genome, analyzing structural variation using short and long reads, extending this into the Alzheimer's. And you can see that as we progress, we go from one sample to hundreds of samples to thousands to tens of thousands. Currently working with the TopMed IRC group to do structural variant calling on over 100,000 whole genomes, which will be the largest such set of its kind. And then you'll see the last step there, going to All of Us Year 1, where that that number drops. But I really want to draw attention to this, that, in the first year of all of us here at the HGSC, we might only be doing 7,000 to 10,000 whole genomes, but they are going to be in this clinical environment. And the important takeaway here is that scale just isn't about how many samples you do. It's also about how you do them. So sure, we've been scaling to more and more research samples, over the last several years, but there's a different type of scaling we're doing now that we're going to be doing these in a CAP-CLI environment and thinking about how we take these all the way through, these samples all the way through to a clinically actionable space. So we have sort of these different ways of scaling, and that's sort of the history and sort of pointing toward the future of what we're working on. And my role as Director of Genome Informatics, one of the hats I wear is thinking about how do we harmonize all of these data. You'll note that on those previous slides where I talked about these large projects, we're working with a lot of sequencing centers, and these data are being generated over a large period of time, and we're bringing data together from a lot of different sources. So, we only have so much control over the quality and the consistency of the data, and we do a lot to think about how do we harmonize those data at scale. And my approach is sort of threefold, or the work that we've done here at the group. Step one is to try to prevent heterogeneity, and I'll talk about some of the mechanisms that we've used and put out to the community to sort of standardize models across the field. You only can do that to a certain extent. You have to be prepared to correct your heterogeneity and adjust for batch effects and deal with that, because they're always going to be there. But finally, you want to embrace the heterogeneity and recognize that different experimental designs, different types of populations, not every batch effect necessarily needs to be removed if that's somehow informative for the type of specific analysis that you're doing. Let your analysis reflect the heterogeneity of the data. So I'm going to walk through a couple of examples how we've dealt with this harmonization at scale, because this harmonization is critical for really building the large sample sets across a diverse, not just a diverse population in terms of data, and what the tools needed to do that are. So thinking about how do you prevent heterogeneity, some of the work we've done in collaboration with the CCDG program was create the NIH functionally equivalent protocol. This is employed by the CCDG, which is currently, again, working on 60,000 whole genomes, the TopMed IRC working on 130,000 plus, the ADSP program, and then the All of Us program just a few weeks ago decided that they'd be using this protocol. This was something developed, again, out of the CCDG in collaboration with TopMed, work led really by the whole group, but Ira Hall and his team at University of or WashU, in St. Louis, really did a lot of great work here. And there's a paper that came out pretty recently that you can look up. But the idea was to get a standardized mechanism of taking a FASTQ all the way through to a CRAM file on the 38 reference, bringing sort of the whole genome programs in the NIH into that 38 space, and also doing so in a way that minimized the file footprint, standardized quality score binning, and provided some features that were amenable to structural variant calling. So all of the big whole genome sequencing programs are on this protocol, and it really makes a lot of the downstream analyses a lot easier when you know that everything was sort of mapped and aligned the same way on the same version. These pipelines are sort of publicly available. If you go read the paper, you'll see that there are Dockers out there. There are apps on all of the different cloud-based providers, including DNA Nexus. And this protocol, it's fast, efficient, and it's definitely where the field is at right now for whole genome sequencing. So that's one way that we've worked to standardize upfront different types of processing. And I'll also point out that you can see here in this results column that doing this actually improved from a cost perspective and from a science perspective a lot of the processing. So it's not just standardization for the sake of standardization. It's standardization because we think we can sort of improve the quality of the data all around. Another mechanism by which we try to standardize is by using robust QC to always assess what's going on. And when we're operating at scale, I think there's a tendency when you want to do QC to downsample things. I'm looking at 100,000 samples. Let me look at 10,000 of them. Let me look at 100 of them. Let me downsample my coverage. So you end up doing these downsamples, and you do your analysis because they're timely or they're expensive. And then, you sort of do the math and you do the very careful thing to make sure that when you scale up, the results you found at the downsampled levels hold. And that's a great model, and we're always going to have to do that. However, given how many smart people there are out there working on fast methods for various genomics tasks, we really should be considering how to do our R&D at the scale. How can we go ahead and make a call set on a large number of variants, look at it and say, okay, we actually need to adjust some parameters and redo that. This idea that we can do our research at our QC at scale is an interesting proposition. One of the tools we've worked with at the HGSC, one of the ones developed here on the team, is XAtlas. It's our small SNV and indel caller. It's rapidly retrainable. It gets from a CRAM to a GVCF in less than a core hour for a 30x whole genome. And, there's some data here that shows that, as you navigate the sort of sensitivity, specificity, time trade-off, that you really can do very fast, high-quality variant calling that allows you to do this type of R&D at scale. And then working with Mike Lin at DNA Nexus on the GL Nexus software, which allows us to go from the GVCFs to a project-level VCF for large numbers of samples in in less than a couple days of processing time, less than 7 core hours per sample. There's lots of mechanisms where having a very rapid small variant caller allow us to iterate quickly and do the type of R&D that's necessary at scale. One example of this is when my team was asked to assess what kind of base level coverage is appropriate in a clinical setting. So here we needed to compare at various base level coverages the ability for a variant caller to accurately access true known variants. Well, the number of samples that we have with high quality truth set variants is much smaller than the scale of things that we are sequencing. So what we did is we took a couple runs of NA12878, which is a very high confidence variant set, and we took deep coverage replicates of these, downsampled them to over 150 different coverages over the whole genome, which gave us a nice spectrum of base coverage across the variants of interest. And we plotted the ability of XAtlas to recover those variant calls at all of these different base coverages. And, we sort of see that around 15x coverage, you can confidently call these types of SNPs. The point here is not the whole exercise of doing the clinical coverage. That's not really what this talk is about. But what I do want to point out is that we were able to run 150 instances of XAtlas and get these variant calls across whole genomes in about an hour because we have, we have a very fast mechanism. It's doing this type of research at scale. It's opening up the types of questions you can ask because you have very fast tools. Another thing that was nice about XAtlas and the ability to run things in a very flexible manner is to adapt to new platforms. When we compared the NovaSeq and the HiSeq, we changed the parameters of XAtlas to sort of do a Pileup, something equivalent to a SAMTools M Pileup, where you're looking at every base and every quality score in every position. But we did this in a very fast manner that also results in a GVCF, which allows us to merge them. And what we saw is that when you start looking at the Pileups in these whole genomes between NovaSeq and HiSeq, you can see a bit of a difference between the TI-TV ratio, which may be something we're still looking into this, that may be related to how the 2-color chemistry differs from the 4-color chemistry. So again, the point here is not the NovaSeq versus the HiSeq comparison. It's the fact that by having a really flexible tool, we're able to dig into the type of innovation that drives, precision medicine and, getting the best technology into the clinic as fast as possible. These types of analyses and being able to tweak our variant caller. Again, another example here is looking at the quality score effects on low allele ratio variants. A lot of these variant callers are trained in a sort of diploid manner. They want to go after things that are roughly a 50% allele ratio, unless you have a somatic caller, which is looking at a different setting. But, you might want to be calling things that are less confident in a germline setting where you're expecting a 50% ratio, but because it might have clinical relevance, you might want to be a bit more sensitive. One of the things that we noticed is that depending on how your quality scores are binned in some of these scenarios, you can see these slight differences in the variant model. and the effect on whether something gets called as a pass or a fail. So the point here being is that as the sequencing technology evolves, as your protocols evolve, the model that you have right now for calling a variance in whatever setting might not necessarily be the right variant calling model for what you would need on a new technology or a new type of experiment. And the ability to retrain that and to do all of the validation very quickly and to understand what's going on very quickly and inexpensively is hugely valuable, because when you're doing things in this clinical setting, you have to be able to go through that validation cycle quickly, so you can ensure that you always have the latest and greatest technology, but it's also up to the high standards of the work that you want to do when you're returning data to patients. The last example I'll give of just being able to do this R&D at scales, again, using these Pileup GBCFs. We looked in the eMERGE panel across 16,000 individuals for sites that might look like they might be a mosaic type variant, where they have allele ratios that are outside of the normal expected 0.5. So we did some filtering and, profiled different variants of how they looked across the whole population to identify putative sites where this might be occurring. But again, the very useful thing here is that because we could rapidly adjust our variant caller and make these Pileup GBCFs, we were able to actually make a mosaic sensitive project level VCF in just a couple of hours using a 32-core instance, again using GLNexus. So again, the idea here is that when we're trying to prevent heterogeneity, we're trying to assess heterogeneity, having these very rapid, scalable tools at your disposal gives you the flexibility to do the innovation you need, and then the ability to rapidly go through the validation that you need for the clinical setting. So it's really marrying the research and the clinical sort of mantras that we need, to move forward into precision medicine. So moving outside of small variants, thinking about how heterogeneity, this is moving away from preventing heterogeneity and assessing heterogeneity to how do we correct or adjust for heterogeneity? And this is starting to get into structural variant calling, large complex variation. And just to go through this, and I apologize if this is basic for everyone, but the idea is that with structural variants as opposed to small variants, the unit of observation, the read, is smaller than the structural variant. So you can't directly observe a structural variant with a short read. You have to infer it from different types of signal, such as read depth, split reads, or read pair discordance, when paired reads don't map in the orientation or the distance that you'd expect. Because structural variant methods are inferential, they're more sensitive to heterogeneity, things like coverage depth, the evenness of coverage, how consistent it is across the genome, the length of the read, the insert size distribution, how close you expect the read pairs to be, and not just the average of that distribution, but the higher moments of that distribution as well, and also the read quality. And this is sort of my example slide, that if you don't account for heterogeneity when calling structural variants, this is an example of some work that we did. 584 samples, we called deletions. We excluded things that were specific to one of the 3 protocols that were used. And even excluding protocol-specific events, we see total segregation of we see total clustering by the protocol. And that includes these 3 replicates that we had, a parent and 2 daughters, that even though it's 3 individuals sequenced 3 times on 3 different protocols, they cluster by where they are, what protocol they were sequenced under, not the individual itself. Now, this is raw data. There was no harmonization. There were different references. But this just goes to show that unless you're accounting for this harmonization up front and you're aware of this, you can have very strong batch effects with structural variation. Our approach to addressing this is the Parliament2 structural variant discovery pipeline, something that we've worked on over the last several years, partnering with DNA Nexus for its design, its execution, its testing, and its deployment. Excuse me. The idea here is that because structural variants aren't one thing and they're sensitive to all these heterogeneities, that different callers are going to leverage the different signals in different ways. So different structural variant callers are going to have different strengths, and we want to leverage the expertise of the field as much as possible. So their different callers might look at different types or size regimes. They might be more sensitive or specific depending on what the goal of the caller is, and they have different compute requirements. So what we've worked on is a single mechanism to fold all of these callers into a single framework, and this is a Docker that's available, that takes in a CRAM file, runs multiple callers, integrates them, and then assigns a quality score based on the profile of that call. What we have found is that multiple callers, obviously, they increase discovery, because the more callers you run, you can see more different types of things. But beyond that, looking at the combinations of callers, saying, okay, well, what different callers were able to support a given event in a sample, that can also inform the quality of that sample. So the idea, again, behind Parliament is that we run multiple callers to leverage their strengths, but also to understand how they interact to inform the quality or the confidence of these individual calls. And before I get into more about the quality of these calls and how we've leveraged this tool at scale, I do want to point out that because of some very clever work integrating these in a single Docker, it runs both on AWS, Google Cloud. We've done extensive testing, although obviously a Docker can run anywhere. But the point is that by putting these together in an appropriate way, we can parallelize them and get several methods for free. You know, there's certain callers or combinations of callers that we're going to take a certain amount of time. We build the right instance on the cloud. We run the other callers in the background. We've also been able to throw in other tools for other things, such as small variant calling and QC metrics. But the idea is that by putting all these together into a single framework, you're running these much faster and less expensively than you would if you were running them individually. I'll also point out that while the field makes the transition from BAMs to CRAMs, if you had to run all of these individually, you know, or in different settings, or you're trying to combine from different places, everyone's doing that work of converting a CRAM to a BAM. A lot of the data that we have now are in CRAM format. So the idea that you're pulling all these together and you're doing all the work in one spot, there's a lot of overhead you're saving as well. And that allows us to execute this at scale. But before we scale up, we really have to focus on how well does it perform by itself on a single sample. And what we have looked at is the genome in a bottle from the NIST organization. They've created a high confidence structural variant set on the HGO2 sample. So we've done the work of taking a 30x sample, short reads, and identifying how well not just the Parliament2 method performs, but how every combination of callers performs in terms of recall of these high confidence events. And one thing to point out is that these high confidence events are generated from extensive, very deep coverage in short reads, long read data, orthogonal technologies. So when we see that the recall for all of these caps out around 71%, we have to keep in mind that a lot of these events were discovered by long reads. So we've done analyses, and if anyone has questions or wants to follow up on this profile, but I'd really encourage everyone to take a good look at the Genome in a Bottle truth sets. That team does great work, and it's a hugely useful resource for everything we do in the research and the clinical community. So what we see here is the recall for different combinations, and you can see that as you start to get 3 or 4 callers, you really start to plateau at about 71% of being able to recall all the events in the truth set, including those that were detected by long reads. Thinking about precision, looking at the different callers and seeing what combinations give you the fewest false positives. These, again, are based on the HGO2 truth set. And what was done here is we use these precision values to assign a quality score to every call that Parliament2 makes based on the type of that call and the combination of callers supporting it. So again, it's not just that multiple callers increase our discovery power, it's also that they help inform the quality of a given call. Before I go into some of these more scaled results, this method has been applied, Parliament2, to all the 1,000 genomes, the about 2,600 low coverage 1,000 genome whole genomes. Before we applied Parliament2, we did remap these to 38 with the NIH functionally equivalent protocol. These calls are available. If you go to the preprint of the Parliament2 paper on bioRxiv, you can find access to these. And to the best of my knowledge, this is the first time that a consistent SV call set on 38 has been done for the 1,000 Genomes project. But even if it's not, it's a great research. It's totally harmonized, and those are available to the community. And just like we did with small variants, thinking about how do we think about QC and harmonizing, one of the things we did with structural variants in Parliament2 was to look at concordance between the NovaSeq and the HiSeq platform and the relative value of the quality scores. And we had 5 replicates, 4 NovaSeqs, 1 HiSeq. We did it at 35 and 50x coverage. And we sort of did filtered and unfiltered. Unfiltered was just taking all the raw calls out of Parliament, ignoring the precision-based quality scores. And then filtered was using only those events with a quality score of greater than 3. And what we can observe here at the top is that the replicate concordance for both NovaSeq versus NovaSeq and NovaSeq versus HiSeq, when you're not using any sort of quality scores, that concordance is lower, and it's very disparate between the 2 types of comparisons. When you do apply these quality scores, that replicate concordance goes up, and it also gets closer. So we're not just improving the quality of the calls, we're also starting to remove some of the batch effects between platforms that we could observe. So that's a double win for us, that we know we're getting more confident calls, and those confident calls are less suspect to what platform the data will run on, which is really important because as we start shifting to the NovaSeq as a field, we don't want to ignore all the HiSeq data. We have to be able to embrace that heterogeneity and just accept the fact that our methods are going to have to adapt. We can't do anything about the raw data itself. And that was sort of our single sample with a high confidence set that we looked at to look at the HiSeq versus the NovaSeq. We also did this at scale. Obviously, we don't have replicates done at the same location for hundreds of samples. What we do have is match samples. And what we did was take 303 HiSeq and 303 NovaSeq and tried to match them in terms of ethnicity, gender, coverage, insert everything. And then we ran Parliament on this to identify whether there would be batch effects. And sure enough, that if you do the principal component analysis, you do see separation by platform in each of the ethnicities, which is perhaps unsurprising given that, the NovaSeq does have a different profile in terms of, the evenness of coverage, especially in certain regions. However, after we apply filtering to these sets, we can see that these differences go away. So we can see that the filtering is appropriate, not just at a single sample level, but also across different ethnicities at a larger scale. So that's a lot of talking about how we QC and how we do the methods, which is all really important because we don't want to jump into actually generating these data until we have an understanding on how we can do so correctly. And the latest example where we're at right now is our cardiovascular disease FREEZE-2 structural variant summary. This is a multi-ethnic population, which is described at the bottom right. We've just identified over 10 million structural variants broken down, described at the top, with about half of them being singletons, which is consistent with what we see on large-scale SNP sets as well. And I'll go into some of these results here about how we did this, but I would like to point out that we were able to generate these structural variant results executing at scale in just about 12 days, and the bottleneck there was really just a job limit, so we didn't break AWS or any of the other systems. But we processed almost a petabyte of CRAMs and 2.3 million core hours in just under 2 weeks. This type of scale work is possible. It is reasonable. You can do whole genome structural variant calling at reasonable prices. And we should just make sure that everyone's on the same page, that these things are feasible and they're becoming increasingly more common. And that's just going to open up a lot more data to the field. But when we generated these results with Parliament, we didn't stop at single sample calling and filtering. Before we got into the genotype phenotype analysis, there's not actually a poster. I just didn't correct that slide. But this is work done by Gu Jun at UT School of Public Health. What we did was take these structural variant calls and analyze them compared to cardiovascular phenotypes. But before we did that analysis, we did do some joint genotyping with CNV. And this is software that takes the calls that we identify in Parliament, profiles all of those positions across the CRAMs for read depth and paired end signals, and then it weights those signals by the extent of multi-method support. The idea being if there's a rare allele somewhere that we might be interested in, but that rare allele because we're not seeing it many times in many samples, but if it is supported by multiple programs, we'll upweight that allele so that signal is more likely to be picked up as a real event. So again, we're not just increasing discovery running multiple methods, we're also improving the type of data we have to improve things like joint genotyping. So what did we do with this joint genotype set? We compared it to risk factors and quantitative traits across cardiovascular disease, interest phenotypes. So all of these were compared, and what we were able to identify were associations. Again, this is just a structural variance set against cardiovascular disease phenotypes. So we observed some novel deletions that are associated with these different genes or clinical markers. And what we've noticed was that as we look at these different ethnicities, we can see that some of these deletions in different events have different effects in different ethnicities. So again, this is the idea that we want to make the work that we do relevant to every individual, every ethnicity, but again, every type of variant, right? If you have a structural variant, we want to make sure that we're doing the type of research that's going to drive the research that will allow that variant to be actionable for your personalized health. So everything that I've sort of described here is what I call better practices. I'm always reluctant to say anything is best practices because we're doing things at unprecedentedly large scale all the time. And the idea that there's one best way to do it, we just don't know. We're doing this as we go. And that's why the R&D at scale is so important and so critical. But these methods have proven successful at scale. They're all publicly available. And I'm always happy to answer questions or if anyone wants to reach out about how we've done this, we're more than happy to share. So now that we've talked about comprehensive genomics and this idea of how do we really look across the whole genome to find all of the variation and do this at scale so it's relevant, get back to this idea of how do we drive that comprehensive genomics back to the N of one personalized medicine? Again, how do we make what we do relevant for every individual carrying any type of variant with any type of phenotype? We really have to think of the full spectrum of genomic scenarios that an individual can be in when they're looking for genomics to help with their health. So as we start to think about the clinic, I think a lot about the sequencing logistics and how do we manage the most important thing when you're dealing with a clinical setting, which is the privacy and respecting the needs of the patient or the participant. This is something that work led by many people here at the center did with the eMERGE network, where there's multiple clinical partners and there's multiple sequencing centers. And the way that we built this to really ensure that we were absolutely moving forward in a way that is appropriate for doing things in the clinical setting is working with DNA Nexus, creating a PHI repository using their features, where we would pull data into this PHI repository, which had all of the compliance that was necessary. We did a de-identification there, and then we pulled all of that down into our local environment for sequencing, which has also had all of the acronym compliance that you would want. We did a PHI join and pushed that back out, excuse me, to our clinical partners. The idea here is that both environments had the appropriate security and compliance, but by sort of boxing off certain steps, you just sort of even further limit any possibility of things getting out. You sort of can separate these so anything on one end doesn't impact things on the other end. And this workflow was totally novel in eMERGE because it was really, as far as I know, one of the first ones where this had to be coordinated among multiple clinical labs, both working within the same program. So this type of infrastructure that we built is really what enables us to start operating at the scale we need. Because, clinical right now, there's a lot of sort of hand-holding that has to be done, or you can't have computers do everything. You need a lot of people who are looking at variants, interacting with patients, doing all of this human work that's incredibly important. And I'm always thinking about how do we capture as much of that as possible to take as much of that time load off of physicians and genetic counselors and patients and everyone involved, so they can focus on the things that they're uniquely qualified to do. And one of the innovations done by here at the HGSC, led by Eric Venner, is the Neptune 2 variant interpretation reporting workflow. The idea is that as you see variants and you start to review them, you automate as much of the annotation and filtering as you can. And when something needs to be reviewed, it's not just that you pass that off for review, is that whatever is done in that review and captured goes back into what we call the VIP database. And that then informs the filtering such that the more variants you see and the more you annotate, the fewer you won't have to do that one again because the automation is going to pick that up. So presumably we're only using the human time to do the new things and we're automating everything else. And that's really going to be the most critical part to scaling a lot of these large programs. I feel like I'm running a little low on time, so I'm going to probably move through the next couple of these pretty quickly. But this is just an example from eMERGE about how the number of cases that are the number of samples that are sequenced, the number of variants that are seen, how does that translate into the number of cases? And this is really something that we're thinking about as we have to scale, is that if we want to do 1 million individuals and we want to start looking outside of protein coding regions or clinically, ACMG 59, as the number of variants that we have to look at scales, we absolutely need to develop the automation and the infrastructure to scale that as well. And it's not just limited to small variants. We've done a lot of work trying to really get high accuracy, single exon resolution, CNV calling and capture designs, and then also extending this again to the whole genome. So again, this is work where, going back to this idea of QC, because of the sequencing strategies, having a method that takes advantage of normalization across multiplex flow cells, for example, and that's what you can see in this preprint of the paper, is really critical to being able to move forward. But there are limitations to short reads only. And one of the things I've been thinking a lot about lately is, this idea of should we do a panel, an exome, or a genome? There's also this component, should we be looking at short reads or should we be looking at long reads? There's a lot of long read technologies out there. They obviously help in structural variants across repetitive regions or seg dupes for small and any type of variant. They do have some challenges associated with them, but this is just one example from Fritz here at the HGSC of an inversion flanked by deletions, where it's very clearly an inversion flanked by 2 heterozygous deletions in the long read data. And, if anyone can look at that short read data and tell me it's an inversion flanked by 2 deletions, please give me a call, I will hire you. But it's not just identifying structural variants. It is also this idea that there's phasing that might be necessary. This is an example of a gene TPMT, where the phasing of 2 variants that are 8,000 base pairs apart impacts the classification that you would then report out. You're never going to get that phasing with short reads alone. So this is just one example of a situation where, in order to have full comprehensive genomics, you might not be able to get there with just one technology. And this is, again, some data from Fritz that shows how long read phasing versus long range linked read phasing, like 10x, how you can get that phasing up by using, again, a different technology. And I think the takeaway is that depending on the type of analysis you want to do, different combinations of short reads, long reads, linked reads are going to be the way to go. You just have to sort of balance that, obviously, with the budget. But the idea is that we're moving toward a world where every genome is a gold genome, and all genomes are going to be clinical. All genomes that we sequence are going to be relevant to personalized health. And we can get to the point where all of them are very high quality. And we think about how we can work towards these whole genome upgrades, taking all of these points into account. And one of these models is thinking beyond a single sample. We have a pilot here at the HGSC, the idea being we've taken a lot of short read whole genome data, we've processed that for structural variants. We've used those short read detected structural variants to identify a subset of samples that would be ideal candidates for long range sequencing. And then once we do the long range sequencing, maybe identify or characterize some complex alleles, we can then, what I'm calling, broadcast those long reads back into the short reads and genotype those long read detected events with the short read data, because genotyping a known event with short reads is a lot easier than detecting it de novo. So this idea of you can combine different technologies, not just in one sample, but across large cohorts, is another way that we can really drive this idea of comprehensive genomics and get the most out of the resources that we have. This is something that, we're absolutely thinking about as an Us Genome Sequencing Center, which is going to be ramping to hundreds of thousands of whole genomes over the next couple of years. And it's not just doing the sequencing, which we know how to scale that. It's really thinking about how do we scale that and do the reporting and get these results back to the participants of all of us in a reasonable manner, looking at whole genome data for, hundreds of thousands of people. So thinking about that scaling and thinking about the security, this is the model that was sort of proposed of us. We merge that with the eMERGE model, where we interact with all of us different groups in the coordinating centers. They handle the PHI intake, but we still do sort of this double de-identification as we work with our partners at Johns Hopkins. Excuse me. The idea being here is that, every group is, treating the data in a way such that it is secure and compliant, and it's lends itself to the automation and the high scale throughput. And this is something that we're really excited about. And the specific things that we're driving in the All of Us program is thinking about FHIR standards and how those deliverables are really going to be standardized across all of the different groups. But the thing I'd like to point out is that none of these projects exist in isolation. I'm not going to go through every one of these points, but all of these projects that we work on talk to each other. For example, thinking about all of the large-scale structural variant calling, we want to reconcile that with the CNV calling in our panel data, and that's going to mean we need to go ahead and pull some of the clinical exome or capture data up onto the 38 reference, and we're going to leverage our work from the CCDG on that functionally equivalent protocol. All of these things interact. And this is the great thing for me working at the sequencing center here in Houston, is that I get to have access to all of these projects and really find the synergy between them and use that to drive, again, the better practices, which is, what pulls us into this, clinical population space. So with that, I do want to leave some time for questions. This work is supported by many, many people and many, many grants. I've highlighted the people who contributed to the slides that I've presented here today. These are all just a great team, especially our collaborators at DNA Nexus. I really appreciate the opportunity to speak today, and I am happy to take any questions.
Thank you, Will, for that informative presentation. We now have time for questions and answers. If you have a question you'd like to ask, please do so now. Just click on the Q&A button at the bottom of the presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the minimal core count you would recommend for utilizing Parliament2 on large data?
So we have optimized this to run on 16 core machines, both on GCP and AWS. And we have found that the disk, 16 cores, somewhere between around 60 gigs of RAM and 200 to 400 gigs of disk attached to that solid state is the appropriate for the speed. That gives us really good performance, and you can scale that disk as needed depending on the size of your cram. But we've played around with that a lot, and the 16 core really is the best balance of time and cost.
Great, thanks.
Yeah,
our second question is, what are your plans for scaling up with each additional year with the All of Us program?
Right, so that's something that we talk with the program about, and they're doing such a fantastic job enrolling people that we're really excited to see that growth. And I'll just sort of put a pitch in. Anyone watching this and anyone can go to All of Us, their website, and sign up and enroll right now. It's just a completely open program. The scaling, there's a combination of whole genome sequencing and whole genome arrays that are going to be done. I think the whole genome sequencing, like I said, we're probably going to be doing a couple thousand, 10,000 the first year, and that's going to ramp really quickly with the goal of having 1 million samples sequenced in 5 years. So we're really expecting to be cranking through many more than 10,000 in the next couple years.
Great. So it looks like we have time for just one more question. Those whose questions we haven't had time for, we'll be following up with you via email. So Will, are there any tools you would recommend outside of XAtlas and Parliament2?
Yeah, absolutely. You know, there's a ton of really great people working out there in so many fields. I would say that one that I really like is SAMplot from Ryan Layer's group for visualizing structural variants. It's really just a smooth program. They've made some nice moves lately on that. Also from Ryan, he's working on the STIX method, which we really like. I've really liked working with the Illumina teams on investigating their Strelka variant caller, especially in the somatic space, and obviously working with the Edico teams on their methods. You know, those FPGAs are really interesting. There's a lot of different, and then all of the constituent programs in Parliament all have great strengths and great development teams behind them. I know people like to be able to just pull something off the shelf and run it, and that's what I hope we created with Parliament. But if anyone has the interest and wherewithal, you really should explore a lot of these spaces. And some of these tools that I've just mentioned, you know, are really a great way to, you know, get your hands on some data and figure out what works best for your experiment.
Right. Great. Well, I'd like to thank once again Will Salerno for his presentation. Do you have any final comments, Will?
No, I just wanted to encourage people to follow up with any other questions as there's more time. Engaging the community like this is how we drive the research necessary to do what we're doing. And I'm really grateful for the opportunity to both DNA Nexus and the group that put this on. Thanks.
All right, thank you. And if you're interested in learning how DNA Nexus provides a scalable cloud environment for clinical data science exploration, analysis, and discovery, please email info@dnanexus.com. or visit dnanexus.com for more information. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing for 6 months from today's live event. You'll receive an email from LabRoots letting you know when this webcast is available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.